All right, everybody. We're going to continue our discussion about physicochemical properties and some of the computational tools we can use to evaluate those properties, and also figure out things like drug likeness. So, is something a drug-like molecule and potentially an orally bioavailable substance? And this is one, I think, our last slide. We were talking about the Lipinski's rule of 5 and the Weber rules, which are sort of complementary rules that sort of determine the same kind of, they predict the same kind of thing, drug likeness. And this is the last example we showed. We're going to see this example again. That's why I'm mentioning it. These are antagonists of a drug target, I guess, neuropeptide Y1. I'm not too sure what this drug target's about, but we're really focusing on drug likeness. And what we found here was that the left structure, kind of their initial lead structure, had sort of bad potency, 2 micromolar, but that overall it was a fairly good molecule in terms of drug-like properties. And when we look at Lipinski's rule of 5, for example, the rule states that the hydrogen bond donors should be less than or equal to 5. Hydrogen bond acceptors should be less than or equal to 10. Molecular weight is less than 500. C log P is less than 5. So that's why, rule of 5 is pretty easy to remember. I still often look it up because it's easy to forget stuff like this. And 1 violation was allowed. Weber rules say that the number of rotatable bonds need to be less than or equal to 10, and the polar surface area needs to be less than or equal to 140 square angstroms. And in this rule, there's no violations allowed. Anyway, just to get us moving again, let's talk about the left compound and the right compound. Left compound, sort of poor potency, but it did pretty much obey the Lipinski's rules and also the Weber rules. In terms of hydrogen bond donors and acceptors, are less than 5 and 10. Molecular weight is less than 500, good. And C log P was a violation at 5.7, but remember we get 1 violation with Lipinski rules. PSA is the polar surface area, and that is 17, which is much less than 140. Rotatable bonds, they calculate 6. I calculated 7. I think I'm calculating them correctly. So this source out of a textbook I think is wrong. Nevertheless, it seems to be in compliance with the Weber rules also. And then in this example, they did some optimization and they changed the molecule, added some stuff, added a couple carbons down here, added a little bit up here, and now the potency is 1 nanomolar, which is great, but now it has very poor oral absorption, and it's not going to be in compliance with these 2 rules. So in terms of hydrogen bond donors and acceptors, it is still in compliance, but the molecular weight is much higher now. It's 591. Previously it was 369. Log P, or we use C log P, which is the computed or calculated C log P. Now it's 7.3, which is much greater than 500. And then the polar surface area is actually not that bad. It's 50 as opposed to 140, but the rotatable bond count, they are counting 14. And they're counting 14 bonds, and I counted 12. I did that last time. But nevertheless, it's still over 10. So based on these parameters, this is neither Lipinski compliant, and also it's not Weber compliant. So kind of explains the poor oral absorption. Okay, that's kind of where we were last time. All right, so next. So we're moving into some of the computer tools for these things. and how to determine it for a chemical structure. Okay, so the first thing we got to do is talk about how we can represent chemical structure in a computer in a very convenient format, and we're going to use something called SMILES, which it's very possible you've never heard of it before. It's a pretty important thing in terms of well, it's been around for a while, we'll say, and it's a great way to represent a chemical structure, even a crazy complicated chemical structure, as just some text, some letters in the alphabet, and we can transmit this by computer really easily. We can copy and paste it really easily. So SMILES is really cool. From there, we'll generate 2D and 3D chemical structures. We're going to predict drug likeness using the Lipinski and Weber criteria that we just talked about. So we'll use a computer to rapidly figure this out, the Lipinski and Weber criteria. Then we're going to actually also use SMILES, these SMILE strings, to predict metabolism of molecules. So we can actually predict the sites of phase 1 and phase 2 metabolism, and also the metabolite structures. So the structures of metabolites. We can predict it by a computer. Super cool. This part will probably be next time. In silico synthesis, which is kind of doing organic synthesis as we understand, but in a computer, which is what we mean by in silico. It's in silico synthesis, and that means kind of computer-based synthesis. We can do really cool things with that. We'll also talk about homology modeling briefly, which is a method to generate a protein structure. If the protein structure does not exist, we can do it by computer. We'll also do docking a little bit. We can't go into depth because these are really complicated things. I just want to introduce them so you'll know what they are at least. Docking, which means docking a small molecule like inhibitor into a protein. And also we'll briefly mention molecular dynamics, which is kind of simulating the protein equilibrating in solution. And it's a really cool thing you can do in drug discovery. Okay, and we may adjust this a little bit more as we go along. All right, so I got my yellow mouse pointer back, which I like to use for PowerPoint, and I will try to remember to do that in the future. Anyway, so let's talk about this SMILES thing. It's a chemical structure representation format that has been used for a while. It's actually created, I think, in the 1980s, which is super surprising to me because somebody that was kind of growing up and going to high school in the 80s, computers were really bad back then, and the internet didn't exist, and I mean, I don't remember, well, I don't think we had things like chemical drawing programs or anything, so it's pretty crazy that people thought of this back then when computers are so bad and preliminary. Okay, so what is it? SMILES is called Simplified Molecular Input Line Entry System. And this is a way to represent a chemical structure in a series of letters, pretty much. And when you think about the diversity of chemical structures, this seems pretty fascinating that you could do this. We'll show some really simple molecules, and we'll show some insanely crazy molecules, and they can all be converted into basically a series of letters and numbers, and it's actually pretty logical, and you can kind of follow it even if you can actually interpret it, is what I'm saying. Not to the extent that a crazy molecule can be interpreted, but maybe easier molecules. Okay, so it was initiated in the 80s, which I'm still fascinated by, and it's really been improved like through the 2000s. I think it's pretty stable now in terms of a lexicon and something like a computer language. It's kind of like a computer language for chemical structures. Okay. Let's talk about ciprofloxacin, which is also called Cipro. It's one of these antibiotic structures. It's a maybe semi-novel antibiotic mechanism. I think it's I forgot what this is inhibiting this second. But anyway, this is a chemical structure, and let's just as an example of what SMILES is, let's we'll sort of step through how the structure is converted into a into a smile string. Okay, so basically some bonds are broken. There's some rings, and some of the bonds in the rings get cut with the little yellow lines. And that's also showing where the cuts are made, and this is all automated. And again, you're not going to be making these strings in terms of like creating them yourself. We're going to use computer programs to help us with this. But yeah, see where the little lines are cut? Those are going to be connection points. And essentially then, now the pieces of the molecule are kind of encoded in color here. And these are all going to be kind of little chains in the smile string. So the green thing is the backbone, and then you got kind of like this sort of orangey thing, and the purple thing, and yeah, and blue, and each of these are going to be kind of components of the smile string. The end result is this thing, and this just, I mean, it's a little hard to follow this, and we'll show a couple easy examples that are easier to follow. But you can kind of see the green thing is sort of the central backbone, and then here and there there's little appendages, and like and those are the differently colored things up top. Okay. The Wikipedia page of smiles is actually pretty good. It's pretty easy to understand, and if you want a little more detail on smiles, that's a place to look. This website is incredibly good. I think this company was maybe involved in the original definition of smiles, daylight.com. And this is on the theory of smiles. So it really explains like kind of how functional groups are identified and built through smiles, and also how ring systems are built. So yeah, there's a lot of theory behind this. Fortunately, it's all computerized now, so nobody really sits around and makes smile strings. We usually use computers to help us with this. Smiles, this is great also. Smiles can be looked up for known compounds. So if it's a known compound, like a known pharmaceutical, like ciprofloxacin, we can actually find it on a website and download it and then use it. And then we can use other tools using smile strings. So PubChem is a really big one, and this is published by the National Library of Medicine. They also do PubMed. PubMed is a kind of a literature searching utility for things in medicine and a lot of chemistry. And also, like if I just have a random molecule that I want to research or use tools with, I can actually draw it myself. And we're going to do that, and you're going to do it in one of your upcoming homework problems too. So I'll show you how to do this and how to use chemical drawing programs. Okay, so here's a couple examples of smiles, and a couple more examples. This is a very simple molecule. This is methyl isocyanate, which is CH3, N double bond C, double bond O. And the smiles of this is pretty straightforward. We're kind of eliminating the H's and the single bonds, and it kind of becomes carbon, nitrogen, carbon, oxygen. So this is the smiles. It's really actually easy. That makes sense, right? We're getting rid of the single bonds, and it's just like carbon, nitrogen, double bond C, double bond O. So that's methyl isocyanate. It gets a little more crazy when we have rings in the molecule. So here is vanillin, which is the molecule that's responsible for the taste and smell of vanilla. It's a aromatic aldehyde with a hydroxy group and a methoxy group. So let's just, for fun, try to interpret this. Okay. Well, we see a C double bond O. Where do you think that is? C double bond O. Oh, well, there it is. It's the aldehyde, right? It's the only C double bond O in the molecule. And then it's got a carbon. That's the capital carbons are, the distinction between capital and lowercase are the capital ones are not aromatic and the lowercase ones are aromatic. Okay, so the capital carbon's there, and then we have this a lowercase c right there. We're also defining this is going to be called number 1, position 1, and that's going to be how the ring is built. Okay, so all the rings get cut. Smiles cuts every ring in the molecule. Okay, so then we have 3 carbons aromatic. 1, 2, 3. Then we have a oxygen. We're ignoring the IH on it, but yeah, that's the phenol. That's the oxygen. Then we have another carbon, aromatic carbon right there. And on that carbon there is a OC. What's the OC? What do you think that is? That's the OCH3. Okay? So that's the methoxy group. And again, we're ignoring the hydrogens. And then we have another carbon. And then we're connected back to position 1. So position 1, which was defined over here, this is how the ring is understood in SMILES. Okay? It's kind of cool. So it's very logical. And again, it's not something we really build these ourselves. We usually use a computer program to help us with it. Isomeric SMILES is a little more complicated. This encodes absolute chiral configuration, which is kind of what we do with like the Kahn-Ingold-Prelog rules. Remember these? And the idea of RNS. So like if we draw an organic molecule that's chiral, we can define RNS, and that defines the absolute configuration. Well, if a molecule is chiral and we use SMILES, it has to do that too, if we have an absolute configuration. And so this concept of isomeric SMILES is kind of like RNS. It's not the same, but it's sort of a similar principle. Okay, all right, so isomeric SMILES, let's just see an example of that. Nicotine is a chiral molecule. And here's the SMILES string. Let's just kind of step through this also. Seeing the C and the N kind of suggests, and the capital C. Capital C, what's the difference between capital and lowercase? The capital is non-aromatic, the lowercase are aromatic. So it's kind of like the right side of the molecule is these left atoms, and the left side of the molecule, the pyridine, is the right side. But we can actually step through all of this, and it is kind of logical. So capital C, capital N, that's the CH3 and the nitrogen, CH3 and the nitrogen. And then that's defining position 1. So nitrogen is, we're going to call that position 1, okay? All right, and then we have 3 carbons, capital CCC, right? Carbon, carbon, carbon. Then we have a chiral center, and that is, so this is how it's doing kind of like what we do as RNS. The rules are a little different, but that defines the chiral center, the little @ symbol. And I think if it's the enantiomer, I believe it's like @, or like it changes there's something about how it does the enantiomer. I think it might be @. Okay, CH, so that's the chiral center, and then that's attached to position 1, so that's the 1 is the bond, and then it's connecting to another carbon, which is it's going to call position 2, and then we have 3 carbons, so it's carbon, carbon, carbon. Those are the aromatic carbons, and then a nitrogen, and there's nitrogen, and then another carbon, and then it's back to position 2. So this bond is the kind of like 2-2 bond. It's between, well, this carbon is the 2 represents where the bond is, I guess, where that bond is, okay? That's kind of how we do rings in SMILES, or how SMILES is defined with rings. Okay, so as a fun exercise, and you're going to do this in your homework, but I'm going to kind of do it a little bit for us also, just to show you how to do it. We're going to look up some molecules just to get their SMILES strings. How do we do that? What are the molecules? Oh yeah, we're going to go to this website, PubChem, NCBI, NLM, NIH, Gov. If you use Google PubChem, it comes right up. Okay, so we're going to try to look up some SMILES strings, and this is a big deal. If it's chiral, if the molecule is chiral, it has chiral centers, we want isomeric SMILES. Otherwise, we'll get something called canonical SMILES. So canonical SMILES doesn't represent chirality. So the thing is, if it's an achiral molecule, all they're going to show is this, is canonical. But if it's a chiral molecule, it's actually going to show both. Okay? And if it shows both, which one are we going to get? We're going to get the isomeric one. Okay? All right, so let's get a couple of these. Desipramine, which is a tricyclic antidepressant. Fluoxetine, which is Prozac. This is a SSRI. Antidepressant. Darunavir is a HIV protease inhibitor. Daptomycin is an antibiotic. And again, we're going to try to get isomeric if we can. Let's just I'll just show a couple of these. Decipramine. All right, so let me see here. PubChem. Okay, so I'm bringing up my browser. First one is decipramine. Okay, so it comes right up. There is the molecule. There may be other variations, which are salt forms, or like hydrochloride, like salt forms, maybe a hydrogen chloride version and stuff. Usually, we're just going to get the top one. Should be sufficient for this course. Okay, so there's the decipramine hydrochloride page, and then we just go down and we can find the there's the canonical smiles. Okay. There's no isomeric smiles for this molecule because there's no chiral centers, okay? But there's the canonical smiles file string for it, okay? And we're going to use these later in a bit. But for now, that's how you find it. Okay, fluoxetine. Let's try fluoxetine. And fluoxetine is Prozac. We can also get the two-dimensional chemical structure of it. It's got an amine and the CF3 benzene, etc. This one I think will also just have the canonical smiles. It is a chiral molecule, but it does not have absolute chirality, and so that's why it doesn't have isomeric smiles. Okay. But if there is isomeric smiles, that's the one we want. All right. Next one was darunavir. Darunavir. Now this is a more complicated chemical structure with obviously a lot of chirality. This is an HIV protease inhibitor, so as we talked about before, it binds to HIV protease and it kind of prevents the scissors from operating in HIV protease. Okay, but this one I imagine will have isomeric smiles. So let's go down, 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 down. There it is. So there's canonical and there's isomeric. So this one we can actually see some of the little at symbols and at. These define the chirality. Canonical does not have that, so this kind of assumes that there's not defined chirality. Another trick, I guess, if we're at the top and we can just Control F, search for smiles, and it comes right down. Okay, so that's how we get down to smiles also. Okay. All right, next one, daptomycin. This is pretty easy. You gotta make sure you spell it right. Now this is a crazy molecule with it's a cyclic peptide with a very complicated structure. And there's a lot of chirality. Each of the alpha positions are chiral, so this will also have a isomeric smiles. So Control F, smiles. All right, there's the canonical, the one we don't want for this one, and here's the isomeric. So this is a big crazy string. It defines that whole molecule, which is pretty amazing. That and this can be generated by a automated computational process. Okay. Okay, so that's where we are now. Okay, so and like I said, isomeric is the desired one. How to draw a molecule to determine smiles. So what if we can't look it up? What if it's a molecule that you made in the research lab and you need a smile string? If you can't look it up, you can't use PubChem, right? So we're going to use a tool that's maintained by the National Cancer Institute and National Institute of Health on this, I guess, Cactus server. And this allows us to draw chemical structures and generate smile strings. Okay, so I can't really assume you guys know how to do this, so I'm gonna show you how to do it. There's a little bit of trick here, so let's try it out. Okay, so I'm gonna copy this cactus string and transfer it. There it is. Okay, so here it is: cactus nci nih.gov. I'm going to make the window a little larger. I just need to see that structure. So let's try this out. Okay. So the trick is going to be, so we're doing, this was also, yeah, this was from your homework assignment number 2. So earlier, it was compound number 5, which was a research compound. That's a non-nucleotide receptor, reverse transcriptase inhibitor. Non-nucleotide reverse transcriptase inhibitor for HIV. And yeah, so we're going to draw this, and you're going to have to draw this. I think the same molecule as part of your own homework, not homework number 2, which we already did, but it'll be I think homework number 5, the new homework. Okay, and so I'm going to go to this button that says Start Structure Editor, and I'm just going to click that, Start Structure Editor. And now we have this kind of structure, this drawing interface. Okay, so I don't know if you can see it totally clearly, so I'll just step through. I'll explain what I'm doing as I do it. I'm going to start drawing this kind of from the left. I'm going to draw the benzene first. So I'm just going to click the benzene button, and there we go. There's our benzene. And then I think it's easy to draw this whole thing as a ring next. And how big is this ring? Because they have different ring sizes, and I can do it all automatically. So how big is the ring? It's 1, 2, 3, 4, 5, 6, 7. So 7-member ring. All right. Let's see. Up top. Okay. Well, there's a 6-member ring. There's a benzene. 7 and 8. Let's just hit the 7 and click here. Now we have a 7-member ring. Okay. And we can draw the oxygen and the nitrogen. I should say that I have a trick I like to do when I draw molecules, which is to kind of fill in the lettered things, nitrogen, oxygen, etc., at the very end, because it's really easy at the end. So I kind of focus on the atomic framework first, and the skeleton, and then I add the oxygens and things like that later. Okay, so let's see. What I'm going to do is I'm going to draw like this ethyl and the benzene. So the way to do that is I go to the single bond utility, and there's the oxygen. Here's going to be the ethyl. We have an ethyl group and a benzene, right? So I'm going to click once and click again. Okay. And so now those are going to be the two things. And the ethyl has an extra carbon, so I just click again. And if I want to draw the benzene, I just click the benzene, and now look at that. Now the benzene looks a little funny. Because it's trying to avoid being connected to the carbon. I'm going to try something different. Oh yeah, another trick we can do is we can undo. So I'm going to see if I can make this look a little nicer. I'm going to undo, and now I'm going to draw the benzene first, and then I'm going to draw that extra carbon. Okay, and here's another trick. Now it connected it to the benzene. If I undo that and click again, nope, it's not working. Oh yeah, what I'm going to do is I'm going to make 2 bonds, and I'm going to delete the first one. That's the trick I'm trying to do. It's a little weird. This drawing program is not perfect, but we can get through our assignment this way. Okay, see what I did? I'll do it one more time. Undo. Okay, so what I need to do is attach the benzene and the ethyl group, right? So if I put the benzene on, how about this? I'm gonna show you the trick I did. So the trick is, say I click once, but I want it to go the other way. I actually want the bond to go to the other direction. The way to do it is click again, and then delete the first bond. See, that's how you do. That's how I do this. Okay, and then I can draw the benzene. All right, so it's a little quirky, but it's doable. You can get stuff done this way. And I'll show one last time the other way I did it, which was first draw the benzene, and then I'm going to click once. It draws it the wrong way. I don't want it to go there. So then I click again, and then I hit the red X to delete the first bond. And that looks cool too. Okay? All right, so we can get through this. The program's not perfect, but we can get this to work. And then we have a double bond to the O. So I think I'll click and then click again. Or you could just use the double bond utility. Okay, what else do we have? A 5-member ring here, so click the 5-member ring and click there. That matches. And then I've got a double bond and then a double bond. Okay, so now I have basically the whole skeleton. Now I'm just going to add my oxygens and nitrogens. So hit the oxygen button. And all right, there's an oxygen, and there's an oxygen, and there is a nitrogen, and we are good. So we drew the molecule successfully. Okay. How do we get the smiles string out of this? I think the trick was hit the smiley face. Ah, there it is. So there's a string, so that we could copy this and paste it. And in your homework assignment, what you're going to have to do is draw these molecules like these, and then you're going to take the smiles and you're going to put it into a Word document. And then you're going to use it later because you're going to do additional stuff with it, like metabolism and other stuff you can do with smiles. Okay? Anyway, that was molecule number 1. Let's do molecule number 2 now. All right, so let's move this thing out of here. Get this out of here. Okay. Back to my PowerPoint. So that was the first molecule. Now we're going to do this molecule. This looks pretty scary. This was from homework number 3. That was the Notch 2/3 inhibitor project. And this is one of the optimal compounds. I think this was the best compound in terms of a potent and selective inhibitor of Notch 2 and 3, which are cancer drug targets. Okay, so let's see if we can do the smiles of this molecule. All right, so now I'm going to move my structure editor back in here. Okay, and I think I can do new or the white box, and it should clear it. Yeah, okay, it clears it. All right, so how are we going to build this? I kind of look at the left. I say, well, that's a nice benzene. Maybe we'll build the molecule from the benzene and then go to the right. So I'll go click on the benzene and click it. Okay, look at that. I wonder if I can do benzene sideways. I just want to see benzene. Nah, I don't. You can only do benzene upright. That's okay. All right, so start with the benzene. And now, how big of a ring are we building? 1, 2, 3, 4, 5, 6, 7-member ring. Find the 7 right there. All right, there we go. There's our 7-member ring. I'm going to also wait on filling in all the other atoms. They're called heteroatoms, yeah. But I'll put the methyl there. Okay. Now we have a double bond O. Uh-oh. I made a double bond in the wrong place. Undo is the little arrow thing here. See the arrow? You'll probably be pressing that a lot. Like I'm sure I will be pressing it a lot as I screw up. We got a benzene down here. There's the benzene. We have a double bond N here. Okay. All right, so we have our double bond now right there. And add a nitrogen. Right. Well, I'll do all those nitrogens later. Okay, so how do we do the stereochemistry here? Because if I do a bond, it's just, that is not a wedge, right? Well, to do a wedge or stereochemistry, there's a little button under the smiley face. A little button under the smiley face. So let's try that and click it. Ah, look at that. So that's the wedge. What if I wanted dashes? I think the trick is just press it again. Okay, so yeah, I can go from wedge to dashes or dashes. Whoa, undo. Dashes back to wedge just by clicking it again. So underneath the smiley face is a stereochemistry button. Okay, And then we have a nitrogen. I'm going to just click and draw that in later. And then we have, click again. Okay, so now we have carbon, nitrogen, carbon, and now we're going to have the double bond O. I actually recommend, I'm going to recommend doing something. I'm going to actually recommend that we draw these things kind of upright like this. Yes, this is going to help with it. Just trust me for a second with the stereochemistry, because I'm going to fill in the nitrogen. Let's fill in the nitrogens now to make this clear. Let's build. We have oxygen, and there's another nitrogen. Okay, you'll see why in a second. But I think it'll be really beneficial if we kind of build the molecule up to the right rather than down to the right. Okay, so now we have a carbonyl. Uh-oh. I want to draw the ketone or the amide, right? Let's just try click. Okay, there we go. That looks pretty good now. Now we have that part. The thing is, the next stereochemistry will be kind of down and wedged. And if what I need to do is build this part kind of going upward. And now we see the wedge, see? That's why I kind of had to build it upward to the right. And so this thing I have to delete now. Okay, so if you're following along, try to get to this point, and then this will really help you if you do it this way. Because otherwise the stereochemistry will be backwards at this position. Okay, so now I'm going to build up. Okay, and then we have down, and then we have. Okay, so now we have oxygen. This will be oxygen, and this thing is another dash dash dash. So going away. Let's use a stereochemistry tool. Okay, so now it's wedged. How do I make it into dashes? We click it again. Cool. All right, so that's cool. And now I need to do, I'm going to do the tert-butyl ester. So it's a tert-butyl ester. So I'm going to do oxygen. And how do we draw the tert-butyl ester? Click again. And then for tert-butyl is carbon with 3 methyls on it. So I'm just going to try clicking, click. Okay, that's a tert-butyl group, OTBU. Okay, now all we have to do is the CF3 stuff. Okay, so let's click and click. So now it's going to be carbon, carbon, carbon. So it's carbon, 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 and 3 fluorines on the end carbon. So now what I'm going to do is draw click, click, click. So now it's going to be carbon, carbon, carbon. Last carbon has 3 fluorines. Now I'm just going to get the F button and go F, 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 F. So now we have that, we did that right, okay? All right. Now I'm going to do it on the other side. Okay, I'm using the mouse wheel. Apparently it lets me zoom in and zoom out a bit. Okay, so let's try the other molecule, the other appendage. So there we go, click, click, and now let's put the 3 fluorines on there. So I use the button and I go click, click, click. Uh-oh, I made a double bond. Undo. Click one more time. Now we have 3 lines, and I'm going to do fluorine, 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 and there we go. We did it. I did it. See if you can do this. And you might, when you're doing this, try to just follow along with my video and make the same mistakes I'm making, and it may actually help you learn how to do this a bit. All right? Okay, so how do we get the final smiles string? And this will be an isomeric smiles because we have defined chirality. It's not going to be canonical smiles. And so hit the smiles button, and then now it's this crazy long thing, this huge string. Okay? I'm gonna save this right now because I may use it in a second. Copy it and open Notepad. I'm pasting it into Notepad, which is a little text notepad in Word or in Office. Or It's a Microsoft tool. Okay, so I just took the string and I put it in this thing. Now, if you're doing your homework, you'd put this in your Word document, right? The smiles. Okay, cool. All right, so back to the PowerPoint. We did smiles for homework 2, compound 5. We also did smiles for homework 3, compound 12. Most recently, we did homework for compound 13B, which was the COVID-19 protease inhibitor. And this should not be that difficult to draw, but there is a little bit that I struggled with. I tried drawing it with that program, the website, and I couldn't get it to work. So we're gonna, I ended up doing this in a different program. And so I will supply this to you for your new homework assignment. Yeah, I used a program called ChemDraw, which I'm not going to get into right now. ChemDraw it's a really cool program, but they make it insanely expensive now. It's almost like $6,000 or something ridiculous. So it's actually very hard to get ChemDraw. We have it in our research lab, kind of like an unofficial license of it. So we're able to use it in our research, and I have it, and most of my chemical drawings, including all of these chemical drawings, are using ChemDraw. But sadly, it's not something you guys have access to. Anyway, one of the nice things ChemDraw does is if I draw a molecule like this, which I did, this is from ChemDraw, I can say make a smiles, and it'll just instantly make a smiles for me in ChemDraw. So that what I'm going to do is give you guys a ChemDraw created smiles version of this molecule. All right. Okay, so next thing. Another thing we can do that you might as well learn right now while we're learning all sorts of things to do with smiles is, say I have a smiles string, and smiles encodes a chemical structure, right? Is there a way I can use a smiles string to identify a compound, identify an organic compound? And the answer is yes, and we can use PubChem. So the PubChem tool will we can input a smiles string and identify a molecule, or close analogs to a molecule. It's a pretty cool thing. Let's try it out. So we're gonna identify a drug from this smile string, because I'm sure you guys can easily look at this and figure out its chemical structure and name, right? It's pretty easy. Let's just look at it. I'm just kidding. It's impossible, right? It's very hard to interpret smiles, and for us as humans to just convert it back to a chemical structure. So what I'm going to do is in my PowerPoint, I'm copying that. Okay, so I just put this in my clipboard. Hopefully that worked. And now what I'm going to do is go to PubChem, Google it. Okay, now we're in PubChem, and I'm going to paste the smiles in, and okay, so it instantly came up with a drug molecule that has that smiles string, and this is oseltamivir. It's also called Tamiflu, which is a antiviral drug for influenza treatment. So another thing about PubChem that's really cool is it actually is kind of like a mini Wikipedia type thing for organic molecules, and so it has all these different things, different names and physical properties, spectral information. So this is a useful tool in your chemistry background as you get do more chemistry. Looking at this, I can actually rotate the three-dimensional structure of it. That's nice. There's all sorts of cool things I can do. Let's try different sticks and wireframe and space filling, so I can look at the three-dimensional structure from different versions. I don't know what Animate does. Oh, Animate will make a little movie for you, so you can rotate molecules around this way. Anyway, so yeah, PubChem is very cool. It's tons of information organized for organic molecules. Here's a mass spec signature, etc. And then it'll show at the bottom related compounds that are maybe similar in structure, and that is nice too. Okay. Okay, so our PubChem search came up with the molecule, and yeah, this is an interesting drug because, as you probably know, bacterial infections, respiratory infections, we treat with antibiotics. Like if you have influenza, though, as a virus, there's not that many drugs that treat influenza actually, as opposed to treating the symptoms. But this is one of them, and this is actually a useful clinical antiviral drug. It inhibits something called neuraminidase. The Wikipedia page actually has a pretty nice description of what this is. That's pretty readable. Essentially, what happens is the influenza is trapped inside a host cell, like a human lung cell or something, and when the virus is trying to leave the cell, there's a whole process that involves sugars on the surface of the cell, and the virus cleaves those sugars and using this enzyme called neuraminidase, and the inhibitor, the antiviral drug, actually blocks their neuraminidase. That's it in a nutshell. There's a lot of figures on this, and probably YouTube videos that show how it works, but it is a pretty cool treatment for influenza. Okay. All right, generation of 2D and 3D structures, and in particular, we're going to do this for SMILES strings. All right, so converting a SMILES string to a 2D or 3D structure. So we just inputted SMILES into PubChem to figure out a molecule, what it is. But we're going to also talk about making 2D and 3D structures. One thing I want to mention real quick, this has nothing to do really with chemistry, how to do a screenshot, because very often, like, you'll want to, you'll find an organic chemical structure on a web page or something, and you want to maybe put it into a file or like a lab report or something. And not everybody knows how to do this, so I do want to mention how do you do a screenshot, like to save a molecular structure. So on a PC, which is all I use, so use Shift Print Screen, and then we can paste the screenshot into a graphics editor. I use a program called IrfanView, which has this little, I guess it's a dead cat, but it's their icon, I don't know. But this is an amazing program. This is like Photoshop for free that is very, very very highly functional graphics editor, and there's other programs like this for the Mac. Oh yeah, so then we crop the image, make it small, paste it somewhere, okay? Like into a Word document or a lab report or something. On a Mac, sadly, I don't know how to help you. There's a lot of YouTube videos on this, but I think it's Shift Command 3 and then something else. Okay, so let's just, as a fun, quick exercise, I'm going to go get the structure of Taxol, paclitaxel, from the Wikipedia page, which has the proper structure of this molecule, and I'm just going to demonstrate how to do a screenshot. Because if you don't know how to do this, you should learn this at some point in your education. So let's just do this real quick, and I'll use the PC method, which is what I'm using. So Wikipedia Taxol. Okay, so paclitaxel, there's the structure. Now there's kind of two ways to do this. One is that I click on this. I'm going to show, what I'm going to do is first show, if I click it again, there's the structure. How do I get this? I'm going to do a screenshot. So on my PC keyboard, I'm going to hit Shift Print Screen. I'm going to open up IrfanView, which is my little editor program. Paste it. So this is a screenshot of my double monitor. And I'm just gonna what I can do is kind of zoom down and get this. Okay, now I have basically the structure of Taxol. So I cropped it. I cropped the image. This is nice because I can paste this like into a Word document. So I opened up Microsoft Word, paste it. Look at that. There, now I have a very high resolution image of Taxol in my Word document or a PowerPoint or whatever. Okay, but let me show one more way to do this. That's kind of cool. So go back. I'll close the open view. Another thing I can do is when I go step back and just click this, like when you've clicked it once, the image looks a little funny like right now. There's all these little checkers. What this is showing is the background is not defined. I can actually copy this image and also paste it into Word. Okay, let's make it smaller. Okay, so now I have two things that look the same, but they're not the same. This was obtained by a screenshot, which is actually pretty good for a lot of things. This was not obtained through a screenshot. It's something called vector graphics. And the cool thing about this is I can actually set the background to like a different color because the background is undefined. So I'm just going to right click, format picture, fill. I can do all sorts of funny, I don't know. See, I can make all sorts of, I can use different backgrounds for my organic structures this way. Anyway, so I just wanted to show that there's kind of two ways that we can use a resource like Wikipedia or any website that has chemical structures. We can either screenshot the image, or in this case, I took the vector graphics version, which allows us to define a different background. Okay, so that was my quick tangent in screenshots. Okay, there's the screenshot method, and here was it's called the SVG method. SVG is called vector graphics, so it's basically the graphical image without the background. It allows us to change the background. So here's another example where I got the SVG image and I was able to define the background, and I did this actually in PowerPoint. Okay, all right. So let's see. Now we're going to convert smiles into a 2D chemical structure. So for this, I found a really good tool at UC Irvine that converts a smiles string into a 2D chemical structure. Until today, I did not know that UC Irvine's mascot is actually an anteater. So yes, they have a crazy, vicious animal like we do. And so we're going to use this. This is going to be the tool that we're going to use. And it lets us take a 2D, smiles and create a 2D chemical image. So let me copy that and we'll open it up. Okay, so now we're on this UC Irvine website. Zoom out a bit. Okay, cool. So I went to this UC Irvine website now, and let's keep going. Okay, so what we're going to do is generate a 2D chemical structure of ritonavir, which is another HIV protease inhibitor, and basically what I did is I found the isomeric smiles from PubChem, and being isomeric, it encodes a stereochemistry. Okay, so this is a pretty nice and crazy long smile string. So I'm going to copy that from the PowerPoint slide. Okay, so I just copied this to my clipboard. Okay, well actually let's go back. All right, so now I'm gonna paste that smiles into this UC Irvine website, and we'll see if it comes up right. So this website is pretty easy to use. These are I'm just gonna erase these smile strings and paste our new smile string. And I think it's just going to work if I just hit generate images. Ah, there it is. Okay, so it actually took the smiles that we fed in and it created a 2D structure. Okay, and I would be able to like copy this image and I think paste it into a Word document. I think it'll work. Why is it not working? Uh-oh. Copy image. Not working. I think I should be able to save it though. Save it and then insert it, or I can always screenshot it, right? Whoops, it's being stupid. So I can always take a screenshot of this. That's why I showed you how to do screenshots, and you're able to like get images based on a smile string. Because I think that's going to be one of the parts of your new homework assignment also, is taking a smiles and figuring out a structure for it. All right, so let's go back to the PowerPoint now. And anyway, so yes, this is the structure of ritonavir that we determined using a smile string. How about a 3D chemical structure? Is there an easy way to convert a smiles string into a 3D structure? Now we did show that if it's in PubChem, we can find it in PubChem and use a 3D. If it's in PubChem, we can look at the 3D structure. But this is a more general tool that we can use for molecules, for any molecule. If I'm given a smile string, well, it could be the chiral one or the non-chiral one. This tool, Galaxy 3D, will always let you determine a 3D structure, which is always really cool. Okay, so we're going to do this on vancomycin, which vancomycin is a very crazy molecule. So there it is. You can probably appreciate the molecular complexity of this huge crazy molecule. And if you look it up on Wikipedia or something, I mean it's got a lot of chiral centers, and it's got quite a structure. This is one of these like last line antibiotics that, like, if every other antibiotic fails and you have a horrible staph infection or something, you'll probably be given this molecule intravenously. Okay, so as you can imagine, this crazy molecule, is this going to be a very simple smile string, or no, it's going to be a very crazy one. So this is the very crazy smile string for vancomycin, and it has every chiral center encoded. This is all the isomeric smiles. The little @ signs are absolute configuration of each chiral center. Okay, and so I'm going to copy it, and in the other window, I just copied this to my clipboard, and let's try this out with Galaxy 3D. So actually what I'm going to do is I can first go to Mole Inspiration. Okay, so I jumped over to the website Mole Inspiration. This is a very cool website. This is a commercial website, but they let you do several things for free, including 3D structure generation. They also let you do a calculation of molecular properties, which are things like C log P, polar surface area, all those things. Remember that? That was from like Lipinski rules and the Weber rules. This tool will allow us to do all that. But for now, we're just going to go to the Galaxy 3D structure generator. Okay. All right, so now I have to get the string. So I'm going to copy and paste the vancomycin smile string into the little box. So let me go to my other window. Okay, so I got it. I got the smile string in my clipboard, and I'm going to paste it right there. Paste the huge string. I can say generate 3D. And this Galaxy 3D structure viewer is actually pretty cool. It does a lot of different things. This is a complicated molecule, so it took a little while to get it, but now we actually have the structure, and we can look at it from different manners. Like I like the tubes. I think, yeah, tubes. This is like it's kind of like it kind of looks like a protein crystal structure or something. We have tubes, we have wires, which is the same kind of thing. We can color the atoms in different ways. I think that polar surface area, no, this is for surface, CPK. Polar surface area. I think this shows the polar groups, carbonyls and nitrogens and things as red. Anyway, this is a pretty neat thing, and given a smile string, we can get a three-dimensional structure. This isn't necessarily the energetically lowest energy structure, but for that, you know, if you want to do that, if you want to actually study the structure and the dynamics of a molecule, you got to use a more complicated program. Probably Spartan. I think Spartan is the one that we use at SF State in a lot of places. Allows you to kind of look at the dynamics of all the bonds and things like that. But this is a quick, easy tool for 3D structure generation. Okay. Anyway, so that shows. Oh yeah, I think from here I'm able to. Can I save this? I think I'm able to save it. Yes, as a image. So I saved the current thing as an image. Let me see if it comes up. Yeah. So I saved that image. It's called a PNG file. It's a graphics file, and then I opened it up with my little OpenView graphics editor, and yes, so we're able to easily create like a static image like this for a homework assignment or a lab report or you know, things like that. All right. Okay, I think we're going to stop again today. I have lecture material for these things, but I have a couple more things I want to get through, and I'm going to wait on that. But the next thing we will do, though, is given smile strings, we'll determine like molecular properties, like the Lipinski parameters and the Weber parameters, and we'll do that on some real molecules. And the next thing after that, we'll take smile strings and we'll predict the metabolic products through phase 1 and phase 2 reactions. Then we're going to get into something even cooler, and this is a little bit related to my research of like in silico synthesis, which is building molecules, building huge libraries of molecules by computer. And then we can kind of like dock them against a protein, and using homology modeling, docking, and molecular dynamics, we can kind of accomplish something called virtual screening, where we're looking for drug molecules by computer. So you know, you have to test them by in the laboratory as well. But if I can screen like 1 million compounds on my computer, and then test like 20 of the best ones, I mean, it can really save you a lot of time in drug discovery. So we're going to get into a little bit of those topics too in this section E. Anyway, for today, I'm going to stop, and then I'll continue on Tuesday. See ya.